Hello, everyone, and thank you for joining us for today's webinar, Harnessing Analytical Development to Empower Process Development for Successful Manufacturing. I'm Leslie Bepko, your host from Bioprocess Online and Life Science Connect. With me today, I have Steven Lal, Lead Scientist in Analytical Development from Samsung Biologics. Just a few housekeeping notes before he gets started. Following his presentation, we'll be holding a live Q&A session. Feel free to submit questions as we go along, and we'll get to as many of those as possible during the Q&A. Any that we can't get to, we'll follow up on after the event. If you run into any issues, please try refreshing your browser first, and if you need additional support, submit a question via the Q&A. Finally, this webinar will be available for on-demand viewing, and you'll receive a link via email as soon as that's available. With all of that said, I'm going to turn it over to Steven to get us started.
Welcome everyone to our webinar today. I'm Steven Lal and I work in the Analytical Development Group here at the Samsung Biologics HQ in Incheon, South Korea. This webinar will highlight our approaches to support your molecule to IND and beyond. The agenda for today's webinar is shown below. Firstly, I will show how we deliver an optimized service to our customers and showcase our in-house capabilities in supporting analytics for cell line development, upstream and downstream processing. Then, I will describe our typical testing panel to support release of drug substance and drug product and extended characterization packages to support structural and physical chemical analyses. Firstly, I'll describe our CDO approach to analytical development. This is an introductory slide to give a top-level overview of our group strengths and capabilities. Our team of highly skilled scientists have an excellent track record of providing end-to-end, high-quality analytics for a plethora of molecule types. Whilst our tried and tested platform processes have been successfully implemented for standard monoclonal antibody products, we recognize the challenges posed by the new modalities that are becoming more prevalent in the ever-changing biopharmaceutical landscape, such as multispecifics, fusion proteins, and recombinant proteins. As you can see from the left-hand side, although a significant percentage of our work has been focused on monoclonal antibodies, we have also supported analytical method development, qualification, and characterization for other modalities, in adherence to ICH QT guidelines where appropriate. This slide demonstrates a typical workflow for the analytical team. Prior to executing lab work, we work with other project teams within the CDO business and the client to understand the analytical requirements that the molecule or the client requires in alignment with the project timeline. Our work typically starts with method development using material supply run material, where we have full flexibility to transfer client methods as required. Following feasibility testing, we perform method qualification and transfer the methods to QC for validation. In general, the same tox material is used to fill an interim reference standard, which is placed on long-term stability and re-qualified annually. Our team also performs long-term accelerated and stress stability studies for non-GMP drug substance and drug product. Finally, we perform structural characterization analyses of the reference standard and biochemical comparability to GMP material. As previously mentioned, our team has experience of working in non-MAB products. There is a drive to more complex proteins, such as bispecifics, single-arm antibodies, FC fusion proteins, to name a few. These pose analytical challenges as we often cannot use our platform methods to assay them. Our analytical team has extensive experience in working with our clients to discuss the methods required to fully understand their molecule. For example, any specific post-translation modification, proteins with multiple binding sites, complex glycan profiles, or multiple N or O glycan sites, and chain assembly. This slide highlights some of the methods that we can perform in order for you to have complete confidence on the start of the development process. We have full in-house capability for all of these methods, so we have direct control over the analytical timelines without relying on outsourcing to other testing houses. Depending on the type of molecule, we perform a range of techniques to demonstrate the product quality during the cell line development, USP and DSP stages. Our newer offerings, sequence variant analysis and HCP-ID by LCMS, which are used to support process development, will be presented later in this webinar. For drug substance and drug product release and stability studies, we perform method development and qualification for the example methods shown on the right-hand side. Methods can be added and removed as appropriate. For instance, for molecules where the chain assembly is critical, for example, bispecifics, reverse phase HPLC could be used. We also have experience in developing dual binding ELISA methods, which may be a preferred option over separate binding ELISAs for bispecific molecules. We also have experience in developing and qualifying cell-based assays such as cell-based proliferation, reporter gene, ADCC, and CDC assays for a range of modalities. The extended characterization panel is critical for IND filing. These methods are applied for reference standard characterization stages and product characterization stages. For example, to assess comparability during process scale-up from 2-liter verification run material, tox batch, and GMP batch scales. The primary structure is fundamental for biologic development, identification, and product homology. Intact mass analysis by ESIMS and peptide mapping are used to confirm the correct amino acid sequence of your molecule and monitor post-translation modifications. One of the most important post-translation modifications is glycans. It is critical to monitor the glycan profile as this can directly impact aspects such as the activity, function, clearance, serum half-life, immunogenicity, and activity. We have developed methods to define the glycan site, occupancy, structures, and profiles for both N and O-link glycans. Higher order structure analysis is critical since changes in the higher order structure can impact the quality, safety, stability, and efficacy, leading to loss of biological function and immunogenicity. Whilst intact mass analysis and peptide mapping can confirm the primary structure, they cannot confirm that the correct disulfide bonding pattern and therefore higher order structure is present. We perform disulfide bond analysis by mass spectrometry as standard for all our programs. In addition, we can understand your molecule's secondary and higher order structures by SEC-MALS, DLS, and CD. Finally, SPR and cell-based methods can be developed as required for your program. It is important to note that this is not an exhaustive list of the services we provide. For example, we also perform in-use stability studies for drug product, force degradation studies, and other analytics to support process development. This slide shows some of the instrumentation used for in-process monitoring, method development and qualification, stability studies, and structural characterization studies. In particular, the highly sensitive ordinary trap fusion LEMOS mass spectrometer in order to quantify protein sequence variant analysis and HCP identification by LCMS will be described further in this webinar. Firstly, our approach to sequence variant analysis will be described. Protein sequence variants are erroneous amino acid substitutions in the primary structure of a protein. These can occur due to mutations at the DNA protein level during DNA replication, clone selection, transfection, and gene amplifications, or amino acid misincorporations during USP or DSP processes. Sequence variants risk cell line stability, give proteins with undesired sequences, and can reduce product quality due to protein misfolding, aggregation, and fragmentation. Although sequence variants are typically present at less than 0.1% in biotherapeutics, the drive for higher titers and productivity can add stress to cell lines, which in turn can lead to errors during protein production. Compared to MAP, the risk of sequence variants can be higher for multi-specific antibodies, fusion proteins, and other modalities, where repeating sequences are more common, particularly as these can be used in flexible linker regions. Samsung Biologics' approach to sequence variant analysis was to develop a method using state-of-the-art LCMS to identify true sequence variants. This has been successfully applied during cell line development and upstream processes. This slide highlights a general workflow for sequence variant analysis. Essentially, the samples are reduced, alkylated, and digested with a protease like a reduced peptide map. After LCMS data acquisition, The data is processed to assign the wild-type peptides and sequence variant peptides by comparison of the retention times and ppm errors. While trypsin is the gold standard for protein digestion, since cleavage gives positively charged ions which aid mass detection by mass spectrometry, there may be cases alone where trypsin is not suitable. For instance, if the theoretical tryptid digest results in long peptides due to the lysine and arginine cleavage sites being far from one another, there may be ionization issues of these large peptides. This, in turn, can impact the detection and quantification of wild-type and sequence variant peptides. This is critical since sequence variants, if present, are generally expected at a low level. Our approach is to use one or more orthogonal digestions using other proteases. In this way, we can have two or more data sets to confirm whether a sequence variant suggested by our software package is real or a false positive. The advantage of our sequence variant analysis method is that we can turn around results very quickly and confidently. Depending on when we decide to implement the sequence variant analysis, this may have little or no impact to our timeline. Our method was developed using NISTmap, a commercially available protein with known sequence variants. As you can see from the chart on the left, our results are consistent with literature values for NISTmap and also shows that our method is suitable to detect even very low sequence variants as low as 0.01% with good MSMS fragmentation as shown by the example NISTmap peptide with the glycine to aspartic acid variant on the right-hand side. Case studies will now be presented to show how our method has been used in process development. The case study on the left shows an asparagine to histidine mutation that was observed during cell line development. Using mass spectrometry, the peptide sequences can be distinguished from one another as not only do they have different masses, our optimized MSMS conditions were able to fragment both peptides to unambiguously confirm the amino acid sequences since we have full B and Y ion coverage for both peptides. This sequence variant was measured at 6% by comparison of the MS intensities of the wild type and sequence variant peptides. This N to H mutation was also confirmed by orthogonal digest. It is important to note, although this sequence variant was present at a relatively high level, typical in-process testing will not necessarily pick up this variant, highlighting the power of our method. In the example on the right, only threonine to serine amino acid substitutions were detected at multiple sites as low-level misincorporations. DNA sequence variants were not detected for this product, and no other sequence variants were detected, suggesting that there was nutrient depletion during upstream processing. These results allowed us to revisit the media composition strategy during upstream processing. This result is similar to the previous misincorporation case. However, samples were tested at different stages of the manufacturing process using CLD, material supply run, and the reference standard samples. The relative percentage of sequence variants increased from CLD to MSR for asparagine to serine variants only. Therefore, we can clearly detect where the manufacturing processes errors can occur. Next, I will present our newest offering, HCP ID by LC-MS/MS. Host cell proteins are process-related impurities produced during the manufacturing of protein drugs. They can be difficult to remove during downstream processing as there can be issues with correlation with the desired protein molecule. They can seriously impact the product quality, efficacy, and safety. HCP ELISA methods are typically of release specification for drug substance. ELISAs can quantify HCPs at a low level and high-throughput methods can be used, but they are unable to identify specific host cell proteins. Knowing what HCPs are present after purification can help develop downstream processing processes. This slide shows some common high-risk HCPs that can be present in biotherapeutics. HCPs can degrade excipients such as polysorbates, degrade the biotherapeutic itself, cleave glycans, or give an immunogenic response. Our method is capable of detecting all of these HCPs. The major challenge of LCMS analysis of HCPs is to overcome the huge intra-sample dynamic range and detect very low abundant, for example, less than 10 ppm residual HCPs. In order to overcome the limitation of the dynamic range by mass spectrometry, sample preparation methods that deplete highly abundant drug substances or enrich low abundant HCPs have been implemented in HCP workflows. Essentially, native digestion methods have also been applied to decrease the dynamic range and improve the intensity and sensitivity for HCPs. HCP-ID methods typically follow a triptych digestion workflow. However, compared to a reduced triptych peptide map for analyzing the drug substance, a much lower triptych concentration under non-reduced conditions is used for this application in order to digest the smaller HCP molecules and leave as much of the drug substance intact as possible. Then, in principle, the undigested drug substance can be removed by precipitation and the supernascent containing the digested HCPs can be isolated and analyzed by LCMS. Absolute quantification methods require a library of HCP standards and known quantities, which can incur high costs and procurement issues. Our method uses a label-free relative quantification workflow rather than absolute quantification. We use a number of internally spiked proteins of known concentrations to cover the range of HCPs in the sample. This is an economical solution to identify and quantify HCPs quickly and at low cost. In this internal case study, we compared verification run material with the first and second tox batches. HCP analysis was performed on these samples as the second tox batch results for HCP ELISA was out of specification. As you can see from the chart, the protein S100 ppm levels from the second batch are much higher compared to the other batches. For the other HCPs in the sample, the levels are comparable across all three batches. Further analysis showed that when comparing pre and post purification material, most HCPs were either completely removed or reduced significantly. However, protein S100 was not effectively decreased for the second tox batch, which is shown in the orange and red colored bars, leading to the out of specification result. It is clear that our method can unambiguously assign a range of HCPs. Knowing specific HCP identities can help optimize purification processes. In summary, the information presented today showcases our full in-house capability to support our clients' requirements of CLD to IND and beyond. We work closely with our customers and within our department to provide high-quality analytics. Finally, our new offerings have been developed to support process development at an early phase. Thank you for listening to our webinar today.
Thank you, Stephen, for a great presentation. If you haven't had a chance to submit your questions yet, please do so now. We'll get to as many of those as we can. And as I said earlier, any that we can't get to, we will definitely follow up with you afterwards. But Stephen, please take a moment, take a drink. Let me get through the questions that we have here. And let's see, let's start with, if either SPA or HCP ID is conducted, what's the impact on the development timeline and cost? And if these extend the timeline and incur additional expenses, are they still valuable?
As mentioned in the slides, both are valuable tools to understand your processes. So depending on when the analysis is conducted for sequence variant analysis, there may be no impact to the timeline at all, although there will be an additional cost in terms of raw materials and man hours. In terms of the timeline, if the SBA analysis is carried out in parallel with the cell line stability stage, typically the SVA data is ready before the cell line stability study is complete, so there'll be no impact to the timeline. For HCPs, for certain applications, HCP levels and the nature for the HCPs may be critical for the type of therapeutic. For instance, ophthalmics often require very low HCP levels. So understanding how the purifications processes can be improved are vital for these types of molecules.
So does Samsung Biologics outsource any of this testing?
The Samsung's analytical team has the in-house capability for the example methods described earlier in the webinar, although we can perform any method as required for our clients. As shown earlier, we have a growing suite of equipment and are continuously developing new methods. Prior to executing a customer project, the CDO team works with the clients to ensure that the correct testing scope and requirements are clearly understood in order to develop the analytical methods required to assay any CQA.
So we have time for one more. Can a client transfer in established analytical methods?
Yeah, that's a great question. So we have experience in developing methods from scratch in-house, or we can tech transfer methods in as per our client requests, depending on where they are with their program. These fine details are discussed at the proposal stage. The purpose of this webinar was to show our analytical capability overall from CLD to IND and beyond. In addition, SVN, HCP-ID analyses are our great new offerings because we can perform them in-house and have strict control over the timeline.
All right, great. Well, Steven, thank you so much. A lot of great information here from Samsung Biologics. If you would like to get in touch with Steven, his contact information is on your screen. So to our audience, thank you so much for joining us. If we didn't get to your question, we will follow up with you afterwards. Thank you for joining us today. Have a great rest of your day.